Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be intimated to you. In the last session, we ended up with MCFA, PUFA, prostaglandins, and compound lipids. In today's session, we will start with very long chain fatty acids. Then we have monounsaturated fatty acids. Then we have modified beta oxidation of MUFA. And lastly, we have polyunsaturated fatty acids, its significance and clinical applications. Morning everyone, let us begin the topic. Here is seen the short chain fatty acids and medium chain fatty acids against long chain fatty acids. Butyric acid in butter and lauric acid in coconut is seen in MCFA, whereas in LCFA, palmitic acid and stearic acid in vegetables and animal fats are seen. In case of MCFA, it is hydrolyzed in stomach, whereas in LCFA, it is not hydrolyzed in the stomach. In case of MCFA, pancreatic acid is not needed, whereas in LCFA, pancreatic acid is essential. In case of MCFA, bile salts are not needed, whereas in LCFA, bile salts are needed. In case of MCFA, inside the intestinal cells, triacylglycerol is hydrolyzed to form fatty acids, whereas in LCFA, inside the intestinal cells, free fatty acids are reesterified to form triacylglycerol. In case of MCFA, it is absorbed directly to the blood, whereas in case of LCFA, it is absorbed into lymphatics and then to the thoracic duct. In case of MCFA, it is oxidized by peripheral cells, whereas LCFA, it is deposited in the adipose tissue. In case of MCFA, carnitine is not required for oxidation, whereas in LCFA, it is required for oxidation. In MCFA, it does not cause atherosclerosis, whereas in LCFA, it causes hypercholesterolemia and hyperatherosclerosis. Coming to very long chain fatty acids. Fatty acid having 20 or more carbon atoms are called very long chain fatty acids. Good examples of VLCA are docosahexaenoic acid and icosapentaenoic acid. DHA, that is docosahexaenoic acid, is synthesized in liver from linolenic acid. DHA is available in large quantities in fish oils. It is required for the development of brain and retina. Decreased quantity of DHA in blood is seen in patients with retinitis pigmentosa. In human beings, the DHA is accumulated in brain before birth and stays there 12 weeks after birth. Outer segments of retinal rods contain high concentrations of DHA, which gives high fluidity to the membrane. This is required for lateral and rotational movement of rhodopsin within the membrane during photoactivation. In this diagram, we can see that rod contains icosapentaenoic acid that is converted into docosahexaenoic acid, which inhibits oxidative stress, which would have produced apoptosis. VLCA is seen in substantial quantities in brain, and components of sphingomyelins are lignoceric acid, nervonic acid, and cerebronic acid. CFAs are partly oxidized to form peroxisomes to smaller fatty acids, which then leave peroxisomes to mitochondria. The peroxisomal oxidation differs from the beta oxidation in that the electrons from FADH2 are directly donated to oxygen to form hydrogen peroxide. So this step does not produce ATP. Peroxisomal production of hydrogen peroxide is one mechanism to kill bacteria by neutrophils. The unwanted hydrogen peroxide is then detoxified by catalase. Catalase is a marker enzyme for peroxisomes. In this diagram, we can see H2O2 converts into 2H2O in the presence of enzyme peroxidase, and remaining hydrogen peroxide is converted to 2H2O by the presence of enzyme catalase. Efficient oxidation of VLCA by peroxisomal enzymes leads to adrenoleukodystrophy, where VLCA accumulates and myelin sheaths are destroyed. It is an X-linked condition. The child usually dies in the first decade of life. In Zellweger syndrome, peroxisomes are not empty or peroxisomal enzymes are not transported correctly into peroxisomes. Zellweger syndrome, adrenoleukodystrophy, and infantile serum disease are together classified as peroxisomal biogenesis disorders. Coming to monounsaturated fatty acids. Human body fat and many vegetable oils contain palmitoleic acid and oleic acid, and mustard oil and rapeseed oil contain erucic acid. Brain contains galvanic acid. The oxidation of unsaturated fatty acids proceeds, as in the case of saturated fatty acids, till the double bond is reached. Thus, palmitoleic acid undergoes 3 cycles of beta oxidation to yield delta 3 cis enoyl CoA with 10 carbon atoms. Here, the double type is cis type. Double bond is cis type. The dehydrogenase cannot act on that bond. In this diagram, we can see that palmitoleic acid undergoes 3 beta oxidation cycles to form cis bond, which again converts into trans bond with the help of enzyme enoyl CoA isomerase. Coming to modified beta oxidation of MUFA, the double bond between third and fourth carbon atoms is shifted to between second and third carbon atoms. It will then undergo second, third, and fourth step reactions by beta oxidation. So, in this cycle, the FAD dependent dehydrogenation is not needed. Thus, in the case of unsaturated fatty acids, the energy yield is less by 1.5 ATP molecules per double bond, because FAD dependent dehydrogenation does not occur at the double bond. Oxidation of very long fatty acids begins in peroxisomes, and once the chain length reaches 20 carbon atoms, they undergo beta oxidation. Coming to polyunsaturated fatty acids. The important polyunsaturated fatty acids are linoleic acid, linolenic acid, and arachidonic acid. They are present in good quantities in vegetable oils such as sunflower oil. They are used to acidify cholesterol, whereby the later can be excreted. So PUFA is in general are antiatherogenic. The other PUFAs belonging to the very long chain fatty acids are timnodonic acid, propanodonic acid, and cervonic acid. They are present in fish oil. They are important for brain development of human brain. Coming to the significance of PUFA. PUFA are seen in vegetable oils. They are nutritionally essential and are called essential fatty acids. Prostaglandins, thromboxanes, and leukotrienes are produced from arachidonic acid. PUFAs form integral part of mitochondrial membrane. In the deficiency of PUFA, the efficiency of biological oxidation is also decreased. They are components of membranes. Arachidonic acid is 10 to 15% of the fatty acids in the membranes. As the double bonds are in cis configuration, the PUFA molecule cannot be closely packed. So PUFAs will increase the fluidity of the membrane. The PUFAs are easily liable to undergo peroxidation, so the membrane contained PUFAs are more prone for damage by free radicals. Here in this diagram, there is oxidative stress which disturbs the membrane. This produces rapid peroxidation. Then this rapid peroxidation brings about damage to mitochondria and cell death. The production of DHG cannot, that is docosahexaenoic acid, from alpha linolenic acid is limited. DHA is present in high concentrations in fish oils. DHA is present in high concentrations in retina, cerebral cortex, and sperms. Coming to clinical significance of PUFA and EFA. Persons with normal diet will not have any deficiency, but those who are on parenteral nutrition or for NOC periods will have deficiency. PUFAs are used for esterification and excretion of cholesterol. PUFAs will decrease the serum cholesterol level. In this diagram, we can see that saturated fatty acids, when given, then that causes decreased LDL receptors and increased cholesterol, whereas MUFAs and PUFAs increase the LDL receptors and decrease the cholesterol. The deficiency of EFA causes acanthocytosis, hyperkeratosis, acrodermatitis, and hypercholesterolemia. It is also connected with acrodermatitis enteropathica, hepatorenal syndrome, and CNS manifestations. Elevated PUFA levels are seen in Zellweger syndrome. DHA levels in blood are decreased in patients with retinitis pigmentosa. The trans fatty acids will compete with EFAs and may increase the EFA suppression and decrease fluidity of membranes. Trans fatty acids decrease the HDL cholesterol and may cause atherosclerosis. Coming to lipid peroxidation. In vitro, peroxidation leads to rancidity of fats and oils. In vivo, the membranes are more liable to attack by free radicals and produce damage to the integrity of the membrane. The naturally occurring antioxidants prevent the lipid peroxidation. Vitamin E and or tocopherol is an important antioxidant in the human body. Here ends our session. Kindly like, share and subscribe to the channel. Thank you.